Hello and welcome, or perhaps maybe welcome back. But yeah, I I pretty much am back again. Less than, or no, a little bit more than 12 hours later. I think if my math is correct. I streamed yesterday or yesterday evening, which I was really happy about. And as I mentioned at the end of the stream, if my sleep holds up, I'll be back again today. I'm really happy to report that I got a pretty good night's sleep last night. Woke up, a quick home workout, kind of some like active recovery day. I'll be back in the gym, my public gym tomorrow. So like yeah, woke up feeling pretty good, and also happy that I got in a good home workout, and still working on my handstands, which I feel like are really coming along nicely. So like yeah, if my sleep continues to hold up, which I'm pretty confident that it will, because I think I know the root of the issue, or I think I've established a routine, like did my best to stick to it, that it has hopefully paid off for the long run. But like yeah, I'm back today with more content, where as usual, I think a lot of science news to cover, um, and I should be back again on Wednesday, where my original goal that I mentioned pretty much over 2 weeks ago is to stream at least like 3, 4 times a week. And that's still my goal moving forward. So I'm going to be trying to do that and accomplish that, just starting this week. And then we'll see how this week goes, and then hopefully carrying that forward. I always try to, at the very least, be somewhat realistic with my goals. I'm almost certainly not going to stream every single day, uh, but I think 3 or 4 times per week, as of right now, is a reasonable goal. Given, let's say, my sleep is not perfect, although it probably never will be, but it's in a much better place than it was, let's say, 3, 4 weeks ago, which I'm really happy about. Where, yeah, I'm still trying to get to the root of the issue, which I think I have. That remains to be seen over the long term. I thought I had it solved 2 weeks ago, and that's when I mentioned that I'll be streaming more, but then everything fell off the rails for no reason whatsoever that I could decipher. But regardless, still always trying to troubleshoot and optimize. But yeah, on today's stream, so yesterday we covered, what did we cover? Oh yeah, pretty much, some bad science practices, and, I guess arguably fraudulent science. And hopefully science correcting itself or exposing sketchy science. And today, as sort of requested by a fellow community member, Zeron Elite, they wanted me to go over some stem cell treatment for diabetes that was approved a couple weeks ago, which is pretty exciting. Lots of development is happening in the stem cell space that I sort of casually follow, but not super closely. But actually, stem cells are being used to treat my own disease, Crohn's disease, for a variety of different uses. Some of the treatments that have been in development have actually failed, or their development has been put on pause. Other ones are still pretty much in the experimental phase, or like generally reserved for very severe cases. So like I do have a personal interest in following stem cell treatment, at least when it comes to my own condition. So I overall follow the research somewhat closely, not super closely. And so like when this came across my newsfeed, I thought it was pretty exciting, because I think it's more or less like a really big step. And we're gonna go back and look at potentially some of the roots of, let's say, the foundational research that took place that resulted in the end in the FDA approving this stem cell treatment. But diving right in, so I covered a news story yesterday about vaping, and I always kind of like try to actively follow the research on vaping, because pretty much an emerging area of research. Even though it's generally regarded as much better than conventional tobacco cigarettes, I think there's still a lot of unknowns when it comes to, as I mentioned yesterday, long-term use of vaping. And I think the overall safety profile of vaping, I think, has maybe, at least in my casual opinion, has been overestimated, or like it may not be as safe as we originally thought it would be. Definitely, or almost certainly, way better than conventional tobacco cigarettes. But again, there's some issues of like, given the fact that it's, marketed as safer and a much better alternative to cigarette smoking, it almost encourages more consumption, as kind of like touched upon here in this article. So you quit smoking with vaping, now what? This is published a few months ago, April of this year. And many people, at least so, vaping appears to be a pretty good, let's say, stepping stone, if not, pretty much has some sort of ability to enable people to quit tobacco cigarettes. But then many people just kind of like stop there, where like, yeah, totally going from cigarettes to vaping, significant improvement in health risk. But then most people just like stop at the vaping, and maybe, stopping at the vaping, not the best thing. Certainly much better, but maybe just transitioning to full quitting of the vaping itself might be like the next step. So every day, Sonia Sharma, a physician assistant, meets people like Natalie H., who is trying to quit vaping. Natalie, a member of the Nicotine Addiction Support Group at the University of California, San Francisco's Fontana Tobacco Treatment Center, switched from traditional cigarettes to vaping, but found the electronic version just as addictive and eventually decided to quit using nicotine completely. I went from being an occasional cigarette smoker, a few a month, to a daily vapor. Right. And I think this is probably more common than it should be. And also not necessarily, at least to me, not necessarily surprising. So said Natalie, who preferred not to give her last name to protect her privacy. Vaping made my nicotine addiction worse, not better. We have people tell us they vape before their feet hit the ground in the morning, said Sharma, who co-leads Natalie's support group at UCSF. Sharma has met individuals who had smoked 4 to 5 cigarettes a day, switched to e-cigarettes to quit smoking, then vaped the equivalent of a pack a day. Others had switched to vapes to quit, but ended up both vaping and smoking again. And others picked up vaping without ever smoking. They want to quit, she said, but are not sure how. Researchers from the National Institutes of Health in 2020 reported that 5.6 million adults in the United States vaped. A little over 57% of people said they started using e-cigarettes to quit smoking traditional cigarettes. Another study in 2021, based on survey data, found that about 60% of e-cigarette users wanted to quit their vaping habit. Vaping has been marketed as a way to help people kick their smoking habit. Research is inconclusive on this claim, but unlike cessation tools like nicotine gums or lozenges, using vapes for cessation is uncharted territory. Vapers lack guidance for how to use the devices to quit, and they have even less direction on what to do if they develop an addiction to the vapes themselves. Right, so again, many people use them as a transition to vaping from traditional cigarettes or conventional cigarettes, and then it's like, well, then what then? They might just stop there, or like vaping in and of itself is now like a new issue. They just replace kind of consumption for one thing for another thing. So a new addiction. Monica Hanna, Assistant Director of the Nicotine and Tobacco Recovery Program at RWJ Barnabas Health Institute for Prevention and Recovery in New Jersey, said she has witnessed a higher level of nicotine addiction in the vapors with whom she has worked. When someone takes a hit from a vaping device, it doesn't generate the burn it would from traditional tobacco, Hanna said. This causes people to take a deeper pull, and when they take a deeper pull, they establish a higher level of nicotine dependence over time. A 2019 study of nearly 900 people published in the New England Journal of Medicine found that smokers who used vapes for cessation were twice as likely to have quit smoking cigarettes than those who used other nicotine replacement therapy. However, 80% of people who switched to vaping were using e-cigarettes a year after they tried to quit smoking. So yeah, again, the issue of like, well, given the fact that they're safer, or like maybe just by virtue of their design, it might encourage, or in fact, it seems to hold true, at least for some people, that it results in just like greater consumption of nicotine. Given that potential for addiction, Nancy Rigotti, director of Massachusetts General Hospital's Tobacco Research and Treatment Center in Boston, said patients must use vapes properly for cessation. That means giving up smoking completely and quitting vapes as soon as patients are sure they will not go back to smoking tobacco. You're going to need to help these people to stop vaping, said Rigotti, who is working with Achieve Life Sciences, a pharmaceutical company developing a prescription drug to treat nicotine addiction from vapes and cigarettes. And many nicotine users who have tried vaping to quit smoking end up becoming dual users, which, I don't know for sure, but I imagine this is arguably more dangerous or has more negative health effects in the long run. It's important to stress that health benefits of switching to vaping only occur if the switch to vapes is complete and permanent. So far, that appears difficult to do for most people who smoke, and in my anecdotal experience, it has not worked, said J. Taylor Hayes, the former medical director of Mayo Clinic's Nicotine Dependence Center in Rochester, Minnesota. Besides challenges in communicating the current evidence, no established method exists to help vapers quit, according to Nargis, Senior Scientific Director of Tobacco Control Research at the American Cancer Society. There are some experimental methods like using social interventions, counseling, and some educational campaigns. Nargis said, little progress has been done in terms of clinical interventions. Unlike cessation products such as gum or nicotine patch, which have clear recommendations for duration of use, similar guidelines don't exist for vapes, in part because the US Food and Drug Administration hasn't yet granted approval of vapes as cessation products. Alex Clark, the CEO of Consumer Advocates for Smoke-Free Alternatives Association, a nonprofit group that supports vaping, said people could vape for longer and still benefit from making the switch from traditional cigarettes. The most important thing is that people start replacing cigarettes with a smoke-free product and continue until they've really switched, said Clark, whose group accepts donations from the e-cigarette industry. Following switching, people are encouraged to continue with the product for as long as they feel necessary. Second. But 2013 guidelines from the FDA advised makers of nicotine replacement therapies, including gums, patches, and lozenges, to include labeling that advises users to complete treatment. According to the agency, if a person feels like they need to use the NRT product for a longer period to keep from smoking, talk to your healthcare provider. Hayes, who is now an emeritus professor at the Mayo Clinic, said he would not recommend patients try vaping as a cessation device, given the availability of more proven techniques such as patches and gums. If a patient insists, vaping could be considered under the medical guidance of a cessation professional. He also advised people purchase products only from large tobacco companies that are likely to have reasonable quality control. Hundreds of vaping devices are on the market, and they are not all equivalent, he says. Right, and this is, I think, also a growing concern of, like, just, at least from what I can tell, I haven't tracked it closely, but just a huge explosion in vaping products and vaping devices. And, I don't know that I have no idea how regulated the industry is. But, I imagine for some companies, if not many of the smaller ones, as sort of hinted at here, not the best quality control. But when an e-cigarette user wants to quit vaping, the guidance might boil down to using traditional tobacco cessation methods like gums and lozenges, because few tools exist to help people with a vaping-specific addiction. The long-term health outcomes of vaping are also unclear, and decades will pass before scientists are able to make conclusions, according to Thomas Eisenberg, PhD, co-director of Virginia Commonwealth University Center for the Study of Tobacco Products in Richmond. I don't think anyone knows what the long-term effects of heated propylene glycol and vegetable glycerin and flavors intended as food ingredients are, especially when these compounds are inhaled hundreds of times a day, week after week, year after year, Eisenberg said. Right. I think this sums it up pretty nicely. Again, I think the research does point to the fact that vaping overall better in comparison to traditional tobacco cigarettes. But again, you are still burning, you're still heating and inhaling, essentially organic solvents or compounds, and almost certainly detrimental to your health long term in some form or another. Maybe not as damaging as tobacco cigarettes, but yeah, if you transition to vaping, at some point you probably want to consider, quitting vaping, which I'm sure is easier said than done. And then just as maybe a preview for future content that I want to cover. This is published today, although I don't have access, a full article, unfortunately. Although I'm going to try and get it through other means. But this is potentially pretty cool, and also like right up my alley in terms of strict biochemistry research, where I've covered protein structure before, and I'm gonna cover it again almost certainly in future content. But this is potentially pretty cool upcoming research, or rather published research, already published research, where this structure, this particular protein, might open avenues, many avenues, when it comes to maybe the treatment of obesity, or as they say here, various metabolism-associated complications. So just as, the byline, the protein UCP1 helps release energy as heat in brown fat. Structures of human UCP1 provide crucial information about its mechanism of action and might aid drug design for obesity and various metabolism-associated complications. What's special about this protein, and what makes this, hotly awaited, is like this protein, as alluded to and highlighted, it's part of brown fat, and brown fat is fairly unique, or stands out as what most people understand as fat, or whatever adipose tissue, is that it burns a lot of energy. Energy in the form of heat. Rather, as it burns energy, it generates heat. And it actually appears to be involved in kind of like, in some aspects in humans, like thermal regulation, or regulating temperature. And it's called UCP1 because it's an uncoupling protein, where pretty much, you are kind of like handling the generation of energy into heat generation, and heat, not really the most useful outside of heat generation, not the most useful form of energy, if not it's completely useless form of energy. And so pretty much like the more brown fat that you potentially have, kind of like the more burning of energy that you can carry out, and you're essentially just like consuming a lot of energy, and that might accelerate weight loss, or rather there are drugs that sort of can promote, let's say, heat burning in your body, that are actually used as weight loss aids, although they're typically illegal or toxic or have other issues. But basically, if you find a way to either stimulate this protein, or the production of this protein, or rather, developing drugs that, allow it to pretty much accelerate its activity, you might be able to, maybe treat certain, as they say, metabolic associated complications. So I'm going to try and get this article and get the associated research, but so far it all appears to be locked away. But with that, getting to the main topic. Published about a month ago, last month. So, FDA approves first cellular therapy to treat patients with type 1 diabetes. So today, the U.S. Food and Drug Administration approved Lantidra, the first allogenic donor pancreatic islet cellular therapy made from deceased donor pancreatic cells for the treatment of type 1 diabetes. Lantidra is approved for the treatment of adults with type 1 diabetes who are unable to approach targeted glycated hemoglobin, average blood glucose levels, because of current repeated episodes of severe hypoglycemia, or low blood sugar, despite intensive diabetes management and education. Severe hypoglycemia is a dangerous condition that can lead to injuries resulting from loss of consciousness or seizures, said Peter Marks, MD, PhD, director of the FDA Center for Biologics Evaluation and Research. Today's approval, the first ever cell therapy to treat patients with type 1 diabetes, provides individuals living with type 1 diabetes and recurrent severe hypoglycemia an additional treatment option to help achieve target blood glucose levels. Type 1 diabetes is a chronic autoimmune disease that requires a lifelong care, including requiring insulin, either through multiple daily injections or continuous infusion using a pump every day to live. People with type 1 diabetes also perform blood glucose checks several times a day to guide the management of their diabetes. Some people with type 1 diabetes have trouble managing the amount of insulin needed every day to prevent hyperglycemia for high blood sugar without causing hypoglycemia. They may also develop hypoglycemia unawareness, where they are unable to detect their blood glucose is dropping and may not have a chance to treat themselves to prevent their blood glucose from further dropping. This makes it difficult to dose insulin. Lantidra provides a potential treatment option for these patients. The primary mechanism of action of Lantidra is believed to be the secretion of insulin by the infused allogenic islet beta cells. In some patients with type 1 diabetes, these infused cells can produce enough insulin so the patient no longer needs to take insulin by injections or pump to control their blood sugar levels. Lantidra is administered as a single infusion into the hepatic liver portal vein. An additional infusion of Lantidra may be performed depending on the patient's response to the initial dose. And Cernu, welcome. Thank you so much for passing by. I hope you're well. That is an excellent question that I am actually going to cover in a bit. But these should essentially be manufactured in a lab, or rather, in a pharmaceutical grade lab. At a pharmaceutical company, since this is actually from a company. Louis 13, welcome. Thank you so much for passing by. I hope you're well. But definitely not mass abortion. But so yeah, I'm gonna go over the roots, or some of the roots of the research that resulted in this FDA approval, because it is a valid question of like, well, a treatment is not all that useful if, you can't scale it up and you can't actually get it to the patients that need it. If you can only provide one dose per year, not really that helpful to the millions of people, unfortunately suffering with type 1 or type 2 diabetes. But this is essentially stem cells grown in a lab that should be grown pretty efficiently, or rather, with enough investment and access to resources, it should be highly scalable on paper. And the research that I'm going to cover in a bit should lend confidence to that. Because the research that I'm going to cover, it's about, I think, 10 years old, or almost 10 years old. And they pretty much close out the research of hey, there's a lot of potential here. There's a lot more steps that need to be done, but this looks like a very realistic way of making these stem cells. And it's approved now. So the safety and effectiveness of Lantidra was evaluated in 2 non-randomized single-arm studies in which a total of 30 participants with type 1 diabetes and hypoglycemic unawareness received at least 1 infusion and a maximum of 3 infusions. This is a fairly small trial. And actually, what's a little bit strange, I'm not going to say that it means anything, but I've actually tried to track down the trials that they're talking about here that resulted in its approval. It doesn't seem like they're published. It seems like basically this research that they're talking about here was only sent to the FDA, which is a little bit, at least in my experience, out of the ordinary. But that doesn't necessarily mean anything, either bad or good. It's just something somewhat noteworthy. But regardless, as I said, I'm going to cover some of the foundational research that has almost certainly contributed to this therapy being approved. But yeah, these are just 2 non-randomized, so it's they are small studies. And, given the fact, even just what they're describing here, there's probably a decent amount of critique that you can put forth on these studies that they're talking about, since that, they're non-randomized, 30 participants. But doing this type of research, with stem cells, with actual human patients with the condition, it's not easy. So where was I? Yeah. So they basically they received infusions of these stem cells, okay? And these stem cells have essentially been engineered so that they produce insulin. So sort of on paper, okay? Specifically when it comes to type 1 diabetes, As a sort of a brief primer, type 1 diabetes, generally considered an autoimmune condition, as in, as a result of your body's immune system. Your immune system is destroying the cells, the insulin producing and secreting cells in your body, and thus you have essentially an insulin deficiency for type 1 diabetics, as opposed to, generally speaking, for type 2, largely like an insulin sensitivity, as in these people are producing insulin, but for a variety of reasons, a lot as a result of metabolic changes for a variety of reasons, these individuals, they essentially have stopped responding to the insulin that they're producing. That's type 2. But for type 1, the cells that actually produce and secrete the insulin, they are actively and continuously being destroyed by their own body. Hence why it's generally classified as an autoimmune condition. Just as my own condition is traditionally, even though technically not an autoimmune condition, but Crohn's disease is often classified as autoimmune because my own immune system is attacking large portions of my digestive system. And that, if I'm not treated and my body is left to do its own thing, like large portions of my digestive system will just be like destroyed. And that is effectively what is happening for type 1 diabetics. Their insulin producing cells are being actively destroyed. But overall, 21 participants did not need to take insulin for a year or more, with 11 participants not needing insulin for 1 to 5 years, and 10 participants not needing insulin for more than 5 years. 5 participants did not achieve any days of insulin dependence. So, pretty stark. Obviously not 100% effective across the board over the long term. Even the ones that responded, there is a variability in their response. But even then, I would say seems pretty promising, or pretty stark, given how current treatment methods available, where for many patients, they have to closely monitor their blood glucose, and they need to either regularly inject themselves with insulin, or they have an actual insulin pump that is kind of whatever, essentially attached to their body, pretty much just as a device that is both monitoring and delivering or administering insulin. So that's effectively what is happening here, but now it's in actual cell form, and you're essentially trying to really mimic as best you can, or as closely as possible currently, what's actually occurring in the human body, trying to replace the cells that have been destroyed in the type 1 diabetic patient. But even then, there were clearly some non-responders. So to not take insulin for 1 year or 1 to 5 years, or even more than 5 years after just a series of injections, that sounds like a really big leap forward when it comes to diabetes treatment. Adverse reactions associated with Lantidra varied with each participant depending on the number of infusions they received and the length of time they were followed, and may not reflect the rates observed in practice. Most common adverse reactions include nausea, fatigue, anemia, diarrhea, and abdominal pain. A majority of participants experienced at least one serious adverse reaction related to the procedure for infusing Lantidra into the hepatic portal vein and the use of immunosuppressive medications needed to maintain the islet cell viability. Some serious adverse reactions required discontinuation of immunosuppressive medications, which resulted in loss of islet cell function and insulin independence. Right, so here's where it's tricky. In some sense, you are kind of doing I'm using the term loosely, but in some sense, you are doing an organ transplantation, or pretty much these cells. Okay, these cells that they're being injected with, they are engineered cells. Okay, so they're not part of their native human body, and so just as if you don't get a donor matched kidney, for example, then if these stem cells are not like, matched to the patient, which they're almost certainly not, given what's occurring, then, for many patients, if not for a significant portion of patients, like eventually their immune system will recognize it. They'll recognize it as foreign and they'll destroy it. Pretty much, just as they're trying to mimic, what a healthy non-diabetic person does in their body. For example, my body, where it's like, yeah, I have insulin producing cells, but they're my cells. Okay, so for these diabetic, type 1 diabetic patients, they're trying to replace those cells, but those cells are not actually part of like their body. They're engineered cells in a lab. And so as they highlight here, for a lot of the patients, it's just like, it's pretty much going to be the same process carrying out, where, the native condition for a type 1 diabetic, it's like, yeah, this is an autoimmune condition. Their own body is attacking, unfortunately, they're attacking their own cells, okay? Even though they're actually native to the person, for whatever reason, for a variety of reasons, largely genetic, they are recognizing their own cells as foreign. But now these patients are being injected with actual foreign cells, actual foreign insulin-producing cells. So yeah, at some point, unfortunately for some of them, for some portion of them, they'll be recognized as foreign and be destroyed. And Rafa is God, welcome. Is all types of... So that's an excellent question. So type 1 and type 2 diabetes, they are definitely treated differently. And just as the terms imply, they are classified differently because the overall, let's say, physiology of the conditions, even though it's under the broad umbrella of diabetes, they're somewhat distinct. Where, as highlighted here, and with this treatment, for type 1 diabetics, it's an autoimmune condition, as in their own immune system is destroying their insulin-producing cells, as opposed to type 2 diabetes, as mentioned earlier, as a result of, let's say, various metabolic changes in the person, they are no longer responsive to the insulin that they are producing, what they term insulin insensitivity. It's like, yeah, the insulin is there, but their body is not really responding to it, or like it's responding much less well to it, as opposed to like for type 1 diabetes, just like there's an absence of insulin. There's an insulin deficiency. And so this treatment here that I'm highlighting is almost certainly or highly unlikely to be effective for a type 2 diabetic. And a lot of the treatments for type 2 diabetic patients, a lot of treatments are, incorporating some lifestyle changes. In addition to, certain pharmaceutical drugs, but lifestyle changes are typically very often recommended in terms of like diet, exercise, etc., etc. So yeah, these adverse events should be considered when assessing the benefits and risks of Lantidra for each patient. Lantidra is approved with patient-directed labeling to inform patients with type 1 diabetes about benefits and risks of Lantidra. And the FDA granted approval of Lantidra to Celltrans Incorporated, which I believe this is, as of right now, pretty small, kind of like startup company, which I imagine, given this approval, they're probably going to have a lot of growth in the next little bit. So let's say covered a little bit more closely, and also from different perspectives. That is also an excellent question. So the answer is, so if they're untreated, they're almost certainly not considered immunocompromised. And if anything, I'm not sure immunosuppressive drugs is part of the treatment for type 1 diabetes. I don't think they are. I believe they just kind of like ignore that aspect of type 1 diabetes. But yeah, as I already highlighted, autoimmune, as in like their immune system is destroying the cells that make their insulin, specifically the beta cells. So what do they say in terms of cause? Yes. Yeah. So the mainstay, regular injection of insulin, manage hyperglycemia. I don't think immunosuppressive drugs are given to type 1 diabetics, just because I don't know for sure, but I don't think that pretty much like once the cells are destroyed, I don't think they're going to like make them anymore. As in, if you stop the immune response, pretty much I think what damage is done is kind of done, I think. I could be totally wrong about that though. But I'm pretty confident that generally speaking, immunosuppressive drugs are typically not given to a type 1 diabetic. And it's just the treatment strategy generally involves just like replacing the insulin. And that's why they have to regularly inject it, or like they have like an insulin pump. So I don't think they would be considered immunocompromised. Just see. Yeah. All the long-term complications pretty much are associated with, blood sugar, or rather, suboptimal management of their blood sugar. And yeah, no surprise, this is pretty common with autoimmune conditions. Of like, hey, if you have one autoimmune condition, you're typically at risk for developing other ones. Let's see. Okay, yeah, so, it looks like, for, let's say, some patients, it is a treatment strategy to involve immunosuppressive, but it seems like it's, the unconventional strategy. So, like, yeah, several other immunosuppressive agents have been trialed with the aim of preventing beta cell destruction. Large trials of cyclosporin, which this is a pretty powerful immunosuppressive agent that typically has pretty severe side effects. Yeah, treatment suggested that cyclosporin could improve insulin secretion in those recently diagnosed with type 1 diabetes. However, people who stopped taking cyclosporin rapidly stopped making insulin, and cyclosporin's kidney toxicity and increased risk. Yeah, as I sort of said, I'm definitely quite familiar with cyclosporin. Where yeah, it might be effective in suppressing the immune system, but it typically has pretty severe side effects, or the potential for pretty severe side effects. So it's like, yeah, it looks like you can suppress the immune system as part of a treatment strategy, but it seems like it's understandably the much less common one, and that if you could just replace the insulin, is the more optimal treatment strategy. But that is a good question. So the U.S. Food and Drug Administration has approved Donesilicel. So this, the actual name of the molecule is that, and then like sort of like the brand name is that Lantidra, a pancreatic islet cell therapy developed from cadaver donors for the treatment of people with type 1 diabetes who are unable to achieve target glucose levels owing to severe hypoglycemic episodes. So it seems like it's this treatment is going to be reserved for individuals that, let's say, are at risk for pretty much well, as they say, severe hypoglycemic episodes. Pretty much, it doesn't seem like it's going to be, for every type 1 diabetic out there, but for, a subset of type 1 diabetic patients. The product is given as a single infusion via the hepatic portal vein into the liver, and a second infusion is given if necessary. And immunosuppression is required to maintain cell viability, just as it is required to support a transplanted kidney or other organ, as these all represent foreign tissues to the recipient. Right, as I sort of highlighted, you are essentially doing, in many respects, organ transplantation. You are injecting foreign cells into the person or in the patient, and so you pretty much want to avoid the patient's immune system from recognizing it as foreign and destroying it. Today's approval, the first ever cell therapy to treat patients with type 1 diabetes, provides individuals living with type 1 diabetes and recurrent severe hypoglycemia an additional treatment option to help achieve target blood glucose levels, said Peter Marks, MD, PhD, director, pretty much part of the FDA. The product was approved despite concerns from the American Society of Transplant Surgeons, the American Society of Transplantation, and an organization of more than 50 transplant surgeons. So it seems like, there has been some pushback, or like not everyone was on board with this being approved. The islets for U.S. Collaborative, whose members argue that cadaver-derived or allogenic pancreatic islets should be regulated as transplanted organs rather than as biologic drugs, as is done in many other parts of the world. As Surnu asked earlier, how would the stem cells be acquired at scale? So I think this answers that question. These cells are being engineered, but they are not being grown, let's say in some sort of like bioreactor. Maybe they're maintained in a lab, but it seems like they're derived from cadavers, and that's why I guess there was like this pushback of like treating them as transplanted organs, because you are getting them from cadavers. So unfortunately, people that have passed away. Lantidra differs from stem cell therapy being developed by Vertex Pharmaceuticals. In the latter, beta cells are grown from allogenic stem cells using a proprietary technology. So far, 6 patients have received the therapy, and it has been successful in all of them to varying degrees, as reported at last week's American Diabetes Association meeting. So this is the one that I'm actually more familiar with, that I'll be covering a little bit of the research about. Where pretty much, not using stem cells or cells from a cadaver and engineering those, but rather pretty much like from more of the ground up, of like using stem cells maintained or like grown in a lab, and then engineering those to produce insulin. So while this is a promising technology with the talk of a cure for type 1 diabetes, it's important to remember that this is very early in the development phase, says Ann Peters, MD, of the University of California, Los Angeles, in her latest column for Medscape. Approval based on small studies with adverse events. Right. So this is directly from like the FDA's, this is an FDA news release. And as they went through, it's based off of 2 non-randomized single-arm studies, a total of 30 participants. So it's non-randomized studies, and small sample size, or small patient sample size. The approval of Lantidra, following a 12-4 vote in favor by the FDA's Cellular, Tissue, and Gene Therapies Advisory Committee in April 2021, was based on 2 non-randomized single-arm studies that included a total of 30 individuals with type 1 diabetes who had hypoglycemic unawareness and who received between 1 and 3 infusions of Donis-Lakel. Again, small studies. So maybe not the highest quality, or almost certainly not the highest quality scientific research or medical research. Insulin independence was achieved at 1 year by 21 participants, so the majority, or essentially 2/3. 11 were still insulin independent at 5 years, and 10 remained so more than 5 years. So about 1/3 of them, it seems like they were able to become insulin independent for more than 5 years, which I would say that's quite a large amount of time. A minority of patients, but still pretty significant. Five participants were unable to discontinue insulin treatment at all. So basically, some non-responders. And yeah, I already went through the adverse events. Anemia, diarrhea, abdominal pain. And most of the participants experienced at least one serious adverse reaction related to the method of infusion and or the use of immunosuppression. So yeah, maybe for like the actual treatment itself, okay, the injection of the stem cells, might not result in side effects. But given the fact that this treatment needs to be coupled with, or very often used in combination with, immunosuppression, well, the immunosuppression may also increase the risk of side effects, or maybe the majority of the side effect risk is coming from the immunosuppression aspect. So yeah, some of these reactions require discontinuation of the immunosuppressive medications, resulting in the loss of islet cell function and return to insulin dependence. These adverse events should be considered when assessing the benefits and risks of Lantidra for each patient. Lantidra is approved with patient-directed labeling to inform patients with type 1 diabetes about benefits and risks of Lantidra. So, U.S. transplant physicians had expressed concern bill introduced. So the transplant surgery organizations had written letters to the FDA, as well as to several other government agencies, to ask that the regulatory framework for Lantidra be shifted from the FDA to the Organ Procurement Transplantation Network and the United Network for Organ Sharing. They also wrote to members of Congress. On June 22nd, 2023, U.S. Senators Mike Lee, Ed Budd, and Marsha Blackburn introduced the Islet Transplantation Bill, which would shift the regulatory framework for cadaveric islets from that of biologic drugs to transplanted organs. Right, so given the fact that, at least for this particular therapy, the Lantidra, that they're derived from cadavers, it seems like there's a push. And I don't really know how to feel about, let's say, the shift, but pretty much to essentially classify them, at least in law, as transplanted organs. They are, I guess, technically speaking, foreign cells coming from another human body. Asked for comment, Peter Witkowski, MD, PhD, the leader of the Islets for U.S. Collaborative, told Medscape Medical News, "We were really happy about the introduction of the Islet bill. Now we're concerned about negative downstream effects of granting a license to a private company for distribution of the cadaveric islets." During the FDA's Cellular, Tissue, and Gene Therapies Advisory Committee's discussion in 2021, several panel members noted that the target patient population for this treatment with the current indication will likely be smaller today than it was when the two studies were initiated. 2004 and 2007, given current automated diabetes technology, such as insulin pumps, continuous glucose monitors, and hybrid closed-loop systems in which the two are linked together as a so-called artificial pancreas that reduces hypoglycemia risk. So it's like, understandably, not everyone is on board with this treatment approved. Multiple things have been flagged. I would say, and this is just my non-expert opinion, it's like I sort of take issue with the fact that they're approving it based off of a small trial that is non-randomized. Basically, my opinion, not the most rigorous research, but carrying out research like this is challenging, and there's a lot of hurdles to overcome. But I can understand why they conducted the studies like this, but then to approve it based off of studies like this, I think it's appropriate to question or criticize. So this brief article here, that's a couple of years old, pretty much is asking that question that Sternu brought up of hey, how are these acquired at scale, and how do we classify them, or how do we view them even in a legal sense of are these just cells in a dish, or are these more closer to organs? And this is published a couple of years, pretty much almost exactly 2 years ago. So, a group of more than 50 experts in the field of pancreatic islet cell transplantation is calling for reclassification of the type 1 diabetes therapy from biologic drug to human organ. If that's your opinion, you're totally entitled to that. I have no doubt that many other people share your position. Not my position, but I understand where you're coming from. And clearly, you know, there's some people that are in line with your position. On April 15th, an advisory panel to the U.S. Food and Drug Administration, and I'd be curious, maybe as a preview of future content that I want to cover. So evolution of a minimal cell. And this is published just a few weeks ago, early July 2023. This is really cool that I am going to cover in depth in a future stream, probably later this week. But pretty much, yeah, as a sort of broad overview and just as a broad preview, I'd be curious, like, where's that dividing line between, a human cell and, like, not a human cell? And, like, hey, what if we can start from, like, really bare bones, and then we can, like, evolve it, and then it becomes a full cell? Because that's essentially what occurred here. They didn't start with a human cell, okay? They started with, if I remember correctly, totally different type of cell. But they pretty much, they hollowed it out, not fully. It's a minimal cell, so it's still a cell. But they pretty much, like, removed a lot, if not, like, a significant portion of the genetic information. And then they let it evolve, or rather they pretty much observed, like, well, given this, like, largely hollowed out cell, but not perfectly hollow, like, would this be subject to evolution? And, like, how many cycles of evolution would it take for it to, like, pretty much get back to or close to its original state? Or even the question of, like, would it actually go back to its original state? Would a lot of the genetic material that was lost, would it be restored if we allowed it to evolve? Like, really, again, not fully hollowed out, but just like a bunch of stuff was eliminated, if not like the vast majority. I'm like, hey, we let it evolve, and a lot of stuff just came back. Kind of like, if humans have the ability to regenerate, themselves or organs, like, how much can, like, we chop away before, like, you're no longer a person? Well, I think you're in the minority. I know you're being sarcastic, but definitely, I'm not of the position that bacteria are human beings, and I think you're definitely in the minority. No advisory panel, I think, is going to be in line with you. But yeah, on April 15th, an advisory panel to the U.S. Food and Drug Administration voted to endorse approval of a biologic license application for the allogenic human islet production, Donislocal, or that Lantidra, developed by Celltrans. The treatment would be intended for people with type 1 diabetes complicated by hypoglycemic unawareness or other issues that result in extreme glycemic lability despite optimized insulin therapy. During the public comment period of the day-long meeting, Peter Witkowski, director of the Pancreatic Islet Cell Transplant Program at the University of Chicago, Illinois, spoke against FDA approval on behalf of the Islets for US Collaborative. And actually, I wonder, like, what exactly this organization is. I should probably just like look off stream and do more research. But I'm curious, you know, just because an institution, you know, has kind of like an official sounding name, doesn't mean it's like actually official or like it's an actual serious institution. But yeah, comprised of over 50 experts in the field, the organization aims to protect islets from commercialization and preserve the human pancreas and islets as public resources for the public good. Witkowski argued that rather than the FDA granting the company an exclusive biologic license application, human islets should be regulated as transplanted organs under the auspices of the Human Resources and Services Administration, with oversight by the Organ Procurement and Transplantation Network and United Network for Organ Sharing, or UNOS. Which, yeah, I'm not so sure about this, personally. That's just my personal opinion. The FDA stance means islet transplants in US fall behind other nations. Human pancreatic islet cells are isolated from deceased donor pancreases and transplanted into the recipient's liver. They require the same immunosuppressive regimens as do all organ transplants. Islets are human microorgans and should be regulated as pancreas and other human organs. Islets are not drugs and they are not cellular therapy, Witkowski emphasized. Right, so like what is an organ? And like what are the limits? Or where do we put the limits? Or like, you know, the drawing the line, you know, a cell and an organ, or like a tissue, a cell, a tissue, and an organ. Those are whatever. Do classify those things. And it's like, you know, if you take just like literally one cell from a pancreas, like that one cell considered a pancreas, it seems like they're kind of like arguing for that. Or, you know, if you take like one liver cell, is that an actual, do we consider that a full liver? Like we do classify these things. Where like, you know, we build up, you know, going from like cell to tissue to organ. To respond. Yeah. I don't want this. Yeah, summarized here. Cells make up tissues, tissues make up organs, and organs make up organ systems. So it's like, yeah, there is like buildup. But like, you know, yeah, like a liver is an organ. If you literally take like one liver cell, you know, not an organ. It's just like one liver cell. Like it's an actual cell. But cells make up tissues, and tissues make up organs. But, you know, their logic is, hey, if you take, like one cell from an organ, well, that, like, new cell that you isolated, that should be considered an organ. Pretty gray area. I said, fair enough. But yeah, islets are human microorgans, as they say. But many parts of the world, including countries in Europe, Canada, Australia, and Japan, regulate islets as organs rather than drugs, he noted. FDA's position that allogenic islets are drugs and require a biologic license application has prevented islet transplantation from becoming a standard of care procedure in the US, in contrast to many other countries, he noted. Witkowski argued that many U.S. academic transplant centers have successfully processed human islets for transplantation in clinical trials without a biologic license application over the past 20 years. But unlike a manufacturer, those institutions are not in a position to obtain such a license. Granting 7 years of marketing exclusivity, he said, would give the manufacturer leverage over availability, cost, and safety monitoring of patients post-transplant. Under HRSA rules, the OPTN or UNOS That's like the United whatever organ something. United Network for Organ Sharing, UNOS. Yeah, UNOS oversees organ transplant programs and monitors patient outcomes following transplantation, but such monitoring isn't mandated for drugs. This raises a safety concern, he asserted. FDA, Celltrans see it differently. There were no responses to public commentary remarks during the meeting per protocol, so Medscape Medical News reached out to the FDA, Celltrans, and the U.S. Department of Health and Human Services. Both FDA and HRSA are agencies within the Health and Human Services, and the OPTN is operated by UNOS under contract with Health and Human Services. A lot of mixing and matching of institutions or organizations. An FDA spokesperson replied by email. The manufacture and transfer of allogenic pancreatic islet cells is not analogous to whole organ pancreas transplantation. Now, we do not agree with the suggestion that pancreatic islets meet the definition of a vascularized human organ. So it's like, well, like they're saying islets are not human. Or like a pancreatic cell is not a full pancreas. The spokesperson also noted, FDA established the regulatory requirements for allogenic islets based on the specific characteristics of that product, its manufacture and intended use, and did so in a transparent manner with input from relevant stakeholders. There is no plan to revise or reconsider this regulatory approach. And Celltrans said, which, understandably, given the fact that they are the manufacturer and own this therapy, they probably don't want more hurdles or more challenges for this being approved, which it has since been approved. Celltrans submitted a biologic license application following federal regulations governing cell therapies. The Code of Federal Regulations on Biologics aims at ensuring safe, high-quality therapies. Celltrans' commitment to safety is evidenced in the filings it has made to the FDA, and will continue to comply with all governmental mandates to maintain safety. The company also said in its email that it aims to offer islet cell transplantation to patients in need as a standard care option. As part of its commitment to facilitate such access, Celltrans has waived all exclusivity rights available to it as part of its application. Moreover, Celltrans is a manufacturer of islets and is not involved in patient care or organ allocation. Celltrans simply processes organs accepted by the transplant center via the UNOS allocation system and provides the islet cell preparation to the hospital transplant center. So it's like, in kind of like the crudest sense, they're accepting organs, okay? And they're processing the organs to isolate the cells or the type of cells that they're interested in, and then they deliver the cells. The HHS press office did not respond by deadline nor after subsequent repeated requests. Based on the discussion at the advisory panel meeting, it's unclear how many patients this issue will affect. Panel members weighed the risks of immunosuppression against the benefit of transplanted islets, given that in recent years, advances in diabetes technology and treatment have reduced the number of patients with hypoglycemic unawareness and extreme glycemic lability. The FDA is expected to announce a decision on Donesical licensure in August 2021. And Tetsu Paul, welcome and shout out to Tetsu Paul, a fitness streamer and Canadian that I am trying to get back into the fitness streaming world. I don't think that worked out the way I wanted it to. But yeah, if you want a great Canadian fitness streamer, Tetsu Paul is definitely the place to go. And hopefully, fingers crossed, in the next month or two, we'll be back with the workout streams. At least that's the plan. I'm pretty optimistic about that. I think I have found a new space for my workout streams. It's just a matter, well, it's largely a matter of convincing the person to allow me to use their space. And I think they're open to it. But I don't want to make any promises that I can't keep. But I really do miss the workout streams. So I really want to, yeah, bring those back as quickly as possible. And definitely Tetsu Paul, if you come to Montreal, you're not obligated to hit me up. But if you totally want to, like, I would love to take you around Montreal. We can go on a Poutine tour as much as you want, because there's obviously a lot of great Poutine places in Montreal, and I know that's your favorite food. And I can eat Poutine all day. I don't, for obvious reasons, but it's like pretty much also one of my favorite foods. Yeah, that'd be great. It could be a lot of fun. But no pressure. Totally up to you. But I'll just say I would love to meet up. So, where are some of the roots of this diabetes treatment that was just approved? But first, maybe a little bit of a primer on diabetes. Or maybe just like a brief overview of like why it's important to hopefully develop new and better treatments for both type 1 and type 2 diabetes. Even though like this particular treatment, it appears to be only or like largely applicable to type 1 diabetics. But both type 1 and type 2 diabetics require much better and improved treatments. But this study here, which is incidence trends of type 1 and type 2 diabetes among use over 10 years, from 2002 to 2012. That makes sense, because I think that's when Otakuthon is. I'm like, yeah, definitely. If you give me some notice, like, I'll totally block off pretty much all the time. Well, obviously, attend Otakuthon, since that's your goal. If you come here, I don't want to keep you from Otakuthon, like your original objective for coming to Montreal and visiting Montreal. But whatever time that you can carve out, yeah, I'll make myself available. I have all kinds of free time for the next little while. But like, yeah, this was in April 2017, so a few years ago. But basically, they're pretty much like trying to track both type 1 and type 2 diabetes over this time period for 10 years, 2002, 2012. And pretty much they conclude the incidence of both type 1 and type 2 diabetes among youths increased significantly in the 2002 and 2012 period, particularly among youths of minority, racial, and ethnic groups. So it seems like for both type 1 and type 2 diabetes, both are increasing, at least during that particular period. And it seems like they're increasing in, as they say, among youths and of minority, racial, and ethnic groups in a particular population. So, all the more reason to develop better therapies, better and improved therapies for these conditions. Where, yeah, it affects millions of people in the United States alone. So, the roots, or some of the roots of this therapy can be traced back to research like this, which was published in around 2014, or late 2014, where the actual treatment that has been approved by the FDA, they're basically, as we just went through, they are injecting cells that are producing insulin into these patients. Basically trying to replace the cells that have been destroyed, or that the patient's immune system has destroyed. and pretty much put it under the category of like a replacement therapy. Hey, these patients don't really have these insulin producing cells, so we will inject, you know, engineered or like harvested and engineered insulin producing cells into these patients. And it seems like for a decent proportion of these patients, the treatment appears to be effective, or at least, you know, it allows them to be insulin independent for some amount of time, from like ranging of 1 year to like 5 years or more. So this type of treatment, you know, its research goes back, you know, about 10 years, if not longer. And this seems like, you know, it was a huge leap forward in the development of this therapy, where, as the title says, generation of functional human pancreatic beta cells in vitro. So basically, I'm a little bit questionable of what you observed, but I'll take your word for it. And like, if the homeless man under the bridge has access to stem cells, and it's hopefully it's actually therapeutic or helps them, but yeah, no judgment on my part. So like, yeah, pretty much as the title implies, they're basically making these cells in a lab, in vitro. They're pretty much like in a dish. As opposed to like the actual therapy that was approved, okay? They're taking cells from cadavers, and they are, you know, processing these cells from the cadavers, and they're pretty much injecting them into patients. So like, not a fully in vitro therapy, as it starts with, unfortunately, you know, a cadaver from a person that has passed away, unfortunately. Oh, and Orange, you welcome. Thank you so much for passing by. Yeah, so beta cells. So beta cells, well, they are part of the pancreas. And good afternoon to you as well. So so the pancreas has multiple functions. But one of its primary functions is to produce insulin. And the beta cells, those are the insulin producing cells. But yeah, they're part of the pancreas, and they're the types of cells found in the pancreas that they make that insulin. Oh, no worries. Thank you so much for passing by. Have a great day of work, if that's even possible. Or rather, I hope you can get some enjoy from your work and you have a great day. But yeah, thanks again. Super appreciated. So like, yeah, they're trying to essentially recreate this particular type of cell from the pancreas. You know, there's the pancreas is whatever, an entire organ composed, if I remember correctly, I could be wrong, but it's composed of multiple different types of cells. But here they're just trying to focus on making the beta cells of the pancreas. Those are the ones that are responsible for making the insulin. And they're pretty much like trying to do it like starting with stem cells, specifically pluripotent stem cells. And then they're trying to like pretty much engineer them to develop into pancreatic beta cells. They're pretty much like starting from kind of like a bare bones cell, and then like trying to get it to develop into a highly specialized type of cell. So, the discovery of human pluripotent stem cells opened the possibility of generating replacement cells and tissues in the laboratory that could be used for disease treatment and drug screening. So pretty much already, like right off the bat, they are sort of hinting at like what was approved for oh, and the Kandar painting, well, Is this the Jack that I know of from the Twitch space? But yeah, four primary cell types: the alpha or A cells that secrete glucagon, beta cells, exactly. So like really trying to focus in on the beta cells. Okay, well, welcome. Thank you so much for passing by. I hope you're well. And also great to see you on Leafy's panel last week. I think it was last Wednesday or last Thursday. And like hopefully I can participate again. If in fact it's a topic that I can speak about. But yeah, already like right off the bat, they are they're hinting at, you know, the treatment that was approved. Not quite the same, because they are starting with, you know, cells from a human body that has passed away, a cadaver. As opposed to here, they want to start off with human pluripotent stem cells. And pluripotent stem cells, okay, they're basically a type of stem cell that they have the potential to develop into multiple different types of cells. Not every single cell, if I remember correctly, you know, there's a difference between pluripotent and totipotent. As in, like, totipotent, they can pretty much like develop into like anything. As opposed to pluripotent, like, you know, there's a lot of potential, but pretty much not as much of an open box. That's already yeah, I've looked this up before. So yeah, pluripotent versus totipotent. Stem cells can divide into all cell types in an organism, as opposed to pluripotent stem cells that can divide into most or all cell types in an organism, but cannot develop into an entire organism on their own. So here, in this research, they are starting off with pluripotent stem cells, not totipotent. So like, it's a lot of potential, but it's not like the full potential, such as you would see in like a totipotent stem cell. Well, thank you so much. And like, yeah, like I said, if it's an actual topic that I can talk about, I will gladly talk about it. But like anime, and a lot of the comic book stuff is probably definitely not my area of expertise. So, and you know, there's a whole, let's say, well, it's a whole other issue of how these pluripotent stem cells are made that I don't really want to get into. But like, yeah, well, you know, let's just say it appears like we have the ability to make these pluripotent stem cells in a variety of different ways. Some ways that people approve of, and some ways that people don't approve of. Okay, if you make that panel, I better be first on the list. And that's when you'll see my debate bro side, you know, yeah, pretty much expose my debate bro side, because I will argue aggressively and angrily for my positions. Recent research has moved the stem cell field closer to that goal through development of strategies to generate cells that would otherwise be difficult to obtain, like neurons or cardiomyocytes. These cells have also been transplanted into animal models, in some cases with a beneficial effect like suppression of arrhythmias with stem cell-derived cardiomyocytes. Right. So like, you know, these pluripotent stem cells, I think as most people know about stem cells, and as we just covered, like there's a lot of potential. Since they can develop into multiple different types of cells, well then, like, yeah, we might be able to generate, as they say, cardiomyocytes, or in this specific research, as we find out, actual pancreatic beta cells that produce insulin. And you have restoration of locomotion after spinal injury with oligodendrocyte progenitors, or improved vision after transplantation of retinal epithelial cells into rodent models of blindness. Right. One of the rapidly growing diseases that may be treatable by stem cell-derived tissues is diabetes, affecting over 300 million people worldwide. So, like, pretty much worldwide, or it's like the equivalent of like if everyone in the United States had some form of diabetes. That's like a huge population. According to the International Diabetes Federation, type 1 diabetes results from autoimmune destruction of beta cells in the pancreatic islet, whereas the more common type 2 diabetes results from peripheral tissue insulin resistance and beta cell dysfunction. Right. As mentioned earlier, that's kind of like, generally speaking, you know, the major or primary distinction between type 1 and type 2 diabetes, where, you know, type 1, as they say, autoimmune, and the patient's immune system is destroying those cells responsible for insulin production, as opposed to type 2, like insulin is being generated. Oh, you switched to the YouTube side, which that should be the ad-free side if you're looking for ad-free viewing. But like, yeah, type 2, you know, insulin is being produced, but essentially the patient has developed insulin resistance, or like is pretty much like non-responsive or like much less responsive to their own insulin that they're producing. So like, you know, sort of like the outcome, or like the end, is sort of the same, in terms of like, you know, for type 1, the insulin is absent, okay? So obviously, since the insulin is absent, the patient is not responding to insulin because it's not there, as opposed to like in type 2, like, yeah, the insulin is actually present, but like the body is essentially not responding to it anymore. Diabetic patients, particularly those suffering from type 1 diabetes, could potentially be cured through transplantation of new beta cells. Patients transplanted with cadaveric human islets can be made insulin independent for 5 years or longer via this strategy, but this approach is limited because of the scarcity and quality of donor islets. So they're specifically actually hinting at the treatment that was approved, you know, about 10 years later. Like I said, this is 2014. Okay. And just recently, you know, last month, we have this cellular therapy. But this cellular therapy, it's derived from cadavers. As they say, deceased donor pancreatic cells. And so they highlight some of the limitations of this type of therapy. Of like, yeah, for a significant portion of patients, it can result in insulin independence, or like, you know, a break from, you know, using or injecting insulin or using an insulin pump. But there's, as they say, the scarcity and quality of donor islets. Oh yeah, absolutely, insulin resistance is a bitch, for sure. The generation of an unlimited supply of human beta cells from stem cells could extend this therapy to millions of new patients and could be an important test case for translating stem cell biology into the clinic. And this research, okay, this is published in Cell. And Cell is pretty much like one of the premier journals in the life sciences, or in pretty much, you know, the world of biology. And you can see, you know, at least some pretty world-renowned research centers involved. You have the Department of Stem Cell and Regenerative Biology, the Harvard Stem Cell Institute, and then you have the Diabetes Center of Excellence. Yeah, Matt Gertler, obviously a whole host of researchers. So, you know, this is pretty large and ambitious research. And so, understandably, a whole host of individuals need to be involved. And it looks like the majority of them, if not like virtually all of them, except for like this person, are affiliated or associated with Harvard or the Harvard Stem Cell Institute. You know, arguably, you know, premier or like, you know, highly regarded and respected research. But yeah, as they say, the generation of an unlimited supply of human beta cells from stem cells could extend this therapy to millions of new patients and could be an important test case for translating stem cell biology into the clinic. So like even this, okay, this would be an improvement on this therapy. If you can just start from human pluripotent stem cells, it will be better than this, because this is, you know, it requires deceased donors, unfortunately. So yeah, this is because only a single cell type, the beta cell, likely needs to be generated, and the mode of delivery is understood: transplantation to a vascularized location within the body with immunoprotection. Pharmaceutical screening to identify new drugs that improve beta cell function, survival, or proliferation is also hindered by limited supplies of islets and high variability due to differential causes of death, donor genetic background, and other factors in their isolation. A consistent, uniform supply of stem cell-derived beta cells would provide a unique and valuable drug discovery platform for diabetes. Additionally, genetically diverse stem cell-derived beta cells could be used for disease modeling in vitro or in vivo. So it's like, yeah, there's clear advantages in pursuing and cultivating, human stem cells, human pluripotent stem cells, for the purposes of generating beta cells, or pancreatic beta cells. Studies on pancreatic development in model organisms identified genes and signals important for the pancreatic lineage, and these have been effectively used to form cells in the beta cell lineage in vitro from human pluripotent stem cells. Definitive endoderm and subsequent pancreatic progenitors can now be differentiated with high efficiency. So this is just like, you know, fancy terminology of like, hey, we can now like do this with like regular routine practice, where like we can pretty much like make these types of cells with not a large amount of barriers. These cells can differentiate into functional beta cells within 3 to 4 months following transplantation into rodents, indicating that some cells in the preparation contain the developmental potential to develop into beta cells if provided enough time and appropriate cues. Right, so like on the pathway of developing fully functioning beta cells. So like they can pretty much generate the precursor to the beta cells, or like the starting point of the beta cell, and then when they transplant that starting point into a rodent, then it looks like it fully develops into a beta cell. But it seems like it takes some time, as they say, within 3 to 4 months following transplantation into rodents. So like, what if we can just go from, you know, that from the human pluripotent stem cell stage to the fully beta cell stage, you know, without the needing for transplantation. Oh, and Zeron, welcome. Thanks so much for passing by. Well, let's see if the YouTube link should be there, I think. Yeah. Where if you want to rewind the VOD on YouTube and start from the beginning, you totally can. Although you can also do it on Twitch, but probably way easier on YouTube. Which is one of the reasons why I also stream on YouTube. Since like the VODs should be there forever and not erased. Also ad-free viewing. And also it should be easier to rewind. So yeah, so like the ultimate goal, so pretty much like the starting point, or like on the pathway to development, is like, yeah, they can start with a human pluripotent stem cell. They can like get up to like, you know, before the beta cell development stage, and they can transplant it into a rodent. And then in the rodent, it seems to develop into a beta cell. It looks like a fully functional beta cell. And now like the ultimate goal, or like, you know, one of the big steps is like, hey, maybe we can bypass transplantation in a rodent, and then we can just go from, you know, human pluripotent stem cell to fully developed pancreatic beta cell without the need for transplantation. And it seems like That's essentially the overall objective of this research. And it also seems like they achieved that. And this is, again, going back in 2014. So, like, 9 years ago. Almost a decade. So, unfortunately, the months-long process these cells undergo in vivo is not understood, and it is unclear whether this process of in vivo differentiation would also occur in human patients. Attempts to date at generating insulin-producing cells from human pancreatic progenitors in vitro have generated cells with immature or abnormal phenotypes. These cells either fail to perform glucose-stimulated insulin secretion in vitro, fail to express appropriate beta cell markers, such as these things here. Not really super relevant, or at least not super critical in order to understand the overall research. They abnormally co-express other hormones like glucagon, fail to function after transplantation in vivo, or display a combination of these abnormal features. So it's like, yeah, basically, they have tried to scale it up to humans, they observe in rodents that, hey, if you just inject the precursors of beta cells into rodents, well, they will develop into beta cells. Well, hypothetically, on paper, that might be achievable in humans, hey, if you can, you might be able to inject the precursor of beta cells into humans, and maybe once inside the human, they will develop into beta cells. What they're saying here is, it seems like that was tried, or something close to that was tried, and it pretty much resulted in negative outcomes, or not overly promising outcomes. So, herein, we report the discovery of a strategy for large-scale production of functional human beta cells from human pluripotent stem cells in vitro, by using sequential modulation of multiple signaling pathways in a three-dimensional cell culture system without any transgenes or genetic modification. We generate glucose-responsive, monohormonal insulin-producing cells that show key features of a bona fide beta cell, including co-expression of key beta cell markers and beta cell ultrastructure. Furthermore, these cells mimic the function of human islets both in vitro and in vivo. Finally, we demonstrate the potential utility of these cells for in vivo transplantation therapy for diabetes. NKX6-1 is a less sensitive but specific biomarker of chromophobe renal cell carcinoma. Would this not be an issue? An excellent question. Even then, well, I would have to do more research, but it's totally a valid question. But even just because something is a biomarker, you can hypothetically, since I don't know the exact role of NKX6-1 in renal cell carcinoma, or chromophobe renal cell carcinoma, which is essentially whatever, kidney cancer. The increase in the biomarker may not itself be inherently an issue. It might just be kind of like a side effect. As in, there might be an increase in the biomarker, but it does not necessarily mean that the biomarker is driving the cancer per se. It might just be as a result of the cancer cells that biomarker is increasing, but it does not necessarily. It's a valid concern, but it does not necessarily mean that increase in the biomarker would drive the cancer. Actually, the best analogy that I can use offhand, okay, is like a CRP, or C-reactive protein, which I'm very familiar with my own condition, where I'll consult the research. That's not how you spell reactive. Okay, so C-reactive protein, it's a marker of inflammation in the body overall, very often used, even just to monitoring risk of heart disease or cardiovascular disease. Well, specifically high sensitivity C-reactive protein. But since my condition is an inflammatory condition, very often, not everyone, not universally, but very frequently, It's used as a biomarker for, let's say, monitoring my own condition, Crohn's disease, or like under the umbrella of inflammatory bowel disease. But just because this biomarker is elevated doesn't actually mean that like it's driving the disease process. As in, you can totally artificially elevate or increase C-reactive protein and have like no effect on the inflammatory process of the disease. It's just that since this is a marker of inflammation, it doesn't mean that if you increase this, that it will drive the inflammation. It's more like just like a byproduct without like dissecting what C-reactive protein is doing or like how it's made. But like it is totally like a valid question and a valid concern. But there's a little bit of like you can potentially disconnect the two, and does not necessarily follow that increasing the biomarker would like drive the cancer or have any effect on the cancer. Just as like there are certain drugs that you can take that will increase C-reactive protein, and it will be, you can totally demonstrate that the inflammation is not getting worse in IBD patients. Oh yeah, sorry for that tangent. I just, very I like getting into the nitty-gritty details. Yeah, exactly. But totally, yeah, that's why it's like I will gladly plead ignorance of like I don't know what NKX6-1 is doing when it comes to chromophobe renal cell carcinoma. Like, but it is a valid concern. Like, if in fact it's a causative factor, then like I can totally see how like if you artificially elevate it, it would make the situation worse. That's why it's like I don't actually know if it's a causative factor. But if it's not, or if it's conclusively demonstrated that it's not, then totally it should not be much of a concern. Obviously, it should be researched, but it's potentially not much of a concern if you're artificially elevating it. Where was I? Right. So like, yeah, what do they show in this research? Or like, why this is published in Cell, okay? And it's affiliated with, an institution like Harvard, with a lot of collaborators or a lot of researchers involved in the research. And so it's like, why is it published in this premier journal? So herein, we report the discovery of a strategy for large-scale production of functional human beta cells from human pluripotent stem cells in vitro. Oh yeah, I already read this. So basically, they're spoiling the plot. I'm not going to dissect the entire paper, just give like most of the highlights. But like, yeah, it seems like, okay, starting from just, a little bit of bare bones stem cells, it looks like they're able to get them to develop into fully functioning beta cells. And it looks like, as they highlight here, that they essentially mimic, actual human beta cells, both in vitro and in vivo. As in, like, outside the organism or inside the organism, it looks like they're, pretty well behaved. And that it looks like there's a lot of potential in using this as an actual treatment for type 1 diabetes. Where like maybe, okay, I haven't followed this research or haven't followed up much with this research, but maybe this is the next generation of this research, or like this is like the next step or the closest next step. That's an improvement. Whereas, as highlighted in this paper, there is a clear challenge with using this therapy because you are using deceased donor pancreatic cells, as opposed to if you're able to just use human pluripotent stem cells, where there might be some issues with generating them, but much less issues with generating them. You could potentially pretty much like have, as they say, an unlimited supply of human beta cells from stem cells. So basically, no production issue. I mean, yeah, I'm not going to dissect all of the methodology and the results, because that's like, I think way too nitty-gritty. But just the highlight, you know, they obviously carry out a lot of experiments. And some of the experiments I'm qualified to dissect and analyze, but some of them I'm totally not. We're just like going through their methodology. You know, I can dissect the experiments in regards to immunohistochemistry, flow cytometry, and even this. But electron microscopy and calcium imaging, I'm in a much not as good position to analyze the results of that type of these types of experiments. I have no experience with electron microscopy. I have experiments with immunohistochemistry, flow cytometry, some with cell culture. Yeah, oh yeah, it is totally, like, very in-depth. Which, you know, I have no problem dissecting it. But obviously, you know, I don't have unlimited time. Or rather, you know, it's a little bit of a shorter stream of, like, 2 to 3 hours. But, like, maybe somewhere down the line. Because, like, I could take the time to, like, understand it and, like, dissect it. And then, like, you know, do a follow-up stream of, like, hey, this is everything that they're doing in great, gory detail. But that's probably going to be, like, an 8-hour stream. I would probably have to, like, invest, like, you know, 12, 24 hours to make sure that I understand everything. And then, like, I'd have to stream for, like, 8 hours to, like, go bit by bit. Which I'm not opposed to doing. It just, like, just takes time, unfortunately. But yeah, they basically engineer the cells, and they get them to develop into, pancreatic beta cells. So, I highlighted pretty much the overall findings. We'll start here in their discussion. We show that functional human stem cell beta cells can be directly generated from human pluripotent stem cells in vitro. So they start in vitro. They pretty much, like, start, you know, sort of like cells in a dish. If I remember my, let's say, Latin correctly, in vitro literally translates to in glass, which I want to make sure that I get that correctly. Give me a definition. Yeah, Latin for in glass. My memory is still intact. Also, having to simplify it down would extend the time frame. Yeah. Which, again, I'm not opposed to. It's just like, I will fully recognize not everyone's cup of tea. And while I personally have an interest in, like, dissecting research like this, like, I love doing deep dives and, like, you know, getting down into the nitty-gritty details. You know, getting dirty, so to speak. But I know that's not everyone's cup of tea. So yeah, the data presented demonstrate that these cells function similarly to primary human beta cells both in vitro and in vivo post-transplantation. These cells can be generated without genetic modification and in large numbers, billions of cells. So, like, they're, as they say pretty explicitly, they're not doing any genetic engineering. Okay. And it looks like they're getting a pretty high yield. They're like billions of cells. And after generating these cells, it looks like once they're transplanted into an organism, I believe here they use rodents or mice, if I remember correctly. It looks like they function, and they function quite well. And so, again, super promising. It looks like they're essentially, again, no genetic modification. They're providing ingredients in the cell culture or in the cellular medium, essentially the growth conditions. And then just through largely or exclusively manipulating the growth conditions, they're able to get them to develop into what looks like, you know, fully functional or close to fully functional beta cells. Though not all of the cells present at the end of the protocol are stem cell beta cells, the percentage of the NKX-1 D-peptide plus cells in the clusters is similar to that found in human islets, and the size of the clusters is comparable, though slightly larger on average than human islets. So it's not perfect, but like it's really close to perfect. This is all accomplished by ex vivo by the addition of defined factors, without the addition of mesenchymal or endothelial cells that normally accompany beta cell development. Although we observe that the global gene expression patterns of stem cell beta cells are more similar to adult human beta cells than to fetal beta cells, our previously produced insulin producing cells, gene expression differences still remain. Oh, you Rich, that Rich. Hey, I'm, that if there's a tribe or crowd out there that likes to get lost in the details, that is my crowd. But I recognize that I'm almost certainly, or we are almost certainly the minority. One possibility is that additional modifications to the culture media or added factors could shift the cells even closer to primary beta cells. So they're hinting at, like, hey, even though they're getting great results here, there's still room for improvement. It seems like they know how to improve it already. Another possibility is that gene expression patterns in cadaveric beta cells result from varied causes of death of the donors. The isolation and recovery process, or shipping, or arise from the influence of adjacent non-endocrine cells. Future studies will include a more detailed analysis of the gene expression differences between stem cell beta and primary beta cells. Two differentiated beta cell markers, so they're just like talking about, well, pretty much like genetic differences or gene expression differences, really getting lost, or like the details. Were not investigated because of the low sensitivity of the probes on the DNA microarray. Looking ahead, it may be possible to use the approach developed here to improve the number of stem cell beta cells formed, as well as alter the differentiation of pancreatic endocrine progenitors to produce all the other endocrine hormone cell types, and reconstruct the proportions found in human islets. So yeah, you have alpha, gamma, delta, epsilon. So it's like, yeah, again. Which, yeah, I don't blame chat. And I know for a fact that there are some people that put on my stream to induce sleep, which I totally respect. And if just some information gets in there through osmosis, I'll take it. But like, yeah, as they're, to summarize, as they're saying, they got pretty drastic and significant results here, but they already see how they can do better. And ways to improve going forward. A preparation of monohormonal cells that respond to a single glucose challenge has been previously reported, but those cells were not shown to express that key beta cell identity marker, NKX6-1, nor function in vivo. Recent work has shown that differentiation protocols that generate higher levels of NKX6-1 lead to better outcomes for the pancreatic progenitor transplants. So, like, it seems like, at least when it comes to generating these types of cells, or as they say, the pancreatic progenitor transplants, it looks like having higher levels of this NKX6-1 is an improvement, or like that is actually better. As you sort of highlight, there might be some sort of collateral damage that will tease out over time, but it seems like increasing this is a good thing in this specific contest. Oh wow, Johnny Scarlet, thank you so much for the raid, and thank you raiders, and shout out to Johnny Scarlet, a fellow education. If I can do that properly. But how was your stream? I hope it went well. I did tune in. Thank you so much for the follow, Admiral Love, and welcome. I did tune in before I went live. I know you were talking about, I think, body positivity and fat acceptance, which I totally have a lot of thoughts as well myself. You know, as someone who's, let's say, involved in health and fitness on Twitch, definitely have some thoughts. Yep, totally. And yeah, thank you so much for the raid, and thank you anyone for tuning in. So I'm Corey Campbell. I'm a science and fitness streamer. I'm a biochemist, and I cover science and news and research. Right now, we're covering some research about some new diabetes therapy that was approved, and we're going over some of the earlier research into the development of these therapies. Specifically, we're talking about stem cell therapy and some of the research about stem cell therapy for the development of treatment for type 1 diabetes. But yeah, take care, be well, and thank you so much for the raid. Super appreciated, super grateful, and welcome, raiders. And yeah, I hope you had a great stream. Yeah, we're going over this paper here, where basically, as highlighted earlier, or as mentioned earlier, you know, type 1 diabetes is largely driven, or if not exclusively driven, by, as they say here, the destruction of beta cells in the pancreas. It's an autoimmune condition, and so for type 1 diabetics, their immune system is destroying their beta cells, and their beta cells are their insulin-producing cells. So one of the therapeutic strategies that's being researched, and that's why I'm covering this research, is like, hey, maybe we can replace those cells, or rather maybe we can engineer those cells or develop those cells and pretty much inject them into the patient, and we can replace their beta cells. The beta cells that were destroyed by kind of like their natural disease process. And so we're covering this research that is pretty much sort of earlier in the research process of going back to 2014, where like this was like a major step where it looks like they were able to really get close to producing these beta cells, just starting from human pluripotent stem cells. And so we're wrapping it up. So where was I? Let's see. Yes. I'm not there. We will start or continue here. Yeah. We note that the insulin detected in serum of transplanted stem cell beta cells was generally lower than that of animals transplanted with cadaveric donor islets on a per cell basis. You know, they're noting some shortcomings of their strategy. Of like, so another strategy that was the one that was actually approved. Okay, going back to what I started off with. Where is it? Too many tabs open. Yeah. So like, yeah, recently, about a month ago, as mentioned, the FDA approved this cellular therapy to treat type 1 diabetes. But this cellular therapy, it's derived from deceased donor pancreatic cells. So that's what they're highlighting here. Of like, their ultimate goal is to generate insulin-producing cells, and it looks like they were able to do that quite successfully. But they compared them to kind of like the other method of pretty much like using the cells from cadavers. And they're saying that, yeah, the amount of insulin that's produced in comparison to those cells appears to be lower. As in, we note that the insulin detected in serum of transplanted stem cell beta cells was generally lower than that of animals transplanted with cadaveric donor islets on a per cell basis. So like, yeah, the cells derived from, you know, cadavers or like deceased donors appear to generate, or it looks like they're generating more insulin than the beta cells that they generated using stem cells. So one possibility is that the other cell types in islets, mesenchyme, endothelial cells, or other endocrine cell types may help engraftment or the vascularization necessary for glucose and insulin exchange from the mouse vasculature. Another possibility is that the slightly larger size of the stem cell beta clusters compared to the human islets that we received may influence relative insulin amount secreted, as it has been observed that smaller islets secrete more insulin per cell than larger islets. So nonetheless, the fact that transplantation of stem cell beta cells consistently results in rapid and glucose-responsive insulin production, and that these cells function to treat diabetes in a mouse model is encouraging. So yeah, even though, they're not performing as well as, the cells derived from, deceased donors, they're still performing really well. Maybe not, it's not perfection, not that perfection exists, but it's still a huge step. And it's like, as they say, super encouraging. The results described here suggest that stem cell beta cells present an opportunity for cell therapy. Limited supplies of donated cadaveric islets and very small amount of human beta cell replication achieved in vitro have severely limited human beta cell supplies to date. This limitation has restricted transplantation options for patients, as well as high throughput drug screening and disease modeling. A single 150-pound patient requires 340 to 750 million transplanted islet cells to effectively resolve type 1 diabetes via islet transplantation. So it's like, I guess that they're pretty much explicitly saying the threshold for, effective treatment, as in pretty much virtually all of the cells in a type 1 diabetic patient, virtually all of the beta cells are destroyed. So if we want to replace them, how many do we have to replace for it to be considered effective? And I guess it varies by weight. And so it's like, yeah, pretty much hundreds of millions of cells, maybe close to 1 billion, I guess, depending on weight. The strategy that we describe here may address this limitation, as it utilizes a renewable cell source and a scalable method, with cells grown 300 million at a time in suspension in a single 500 ml flask. Interesting. Thus, 1 or 2 flasks may be sufficient for treatment of a patient. An additional clinical advantage of stem cell beta cells compared to previously proposed therapies using pancreatic progenitors is that these cells do not require an extended post-transplantation period before observing insulin production. Immediate insulin production makes it theoretically possible to reduce patient exogenous insulin requirements from the first few days post-transplantation. So it seems like there is essentially a quick turnaround time. So, these cells also present a new opportunity for beta cell studies. A single flask could provide enough cells for screening 30,000 compounds in a 384 well format seeded with 10,000 cells per well. Unlike primary human beta cells, stem cell beta cells can also be generated from pluripotent cells of any desired genetic background. Human-induced pluripotent stem cells, which function like can't remember what this acronym is, but another type of stem cells, can be generated from any fibroblast or other somatic cell types through introduction of a small set of pluripotency genes. Human-induced pluripotent stem cells. Actually, I'm highlighting a little bit of whatever. I'm nitpicking. This is like a mistake or like a typo, because the HIPSC, it's redundant. The HIPSC stands for human-induced pluripotent stem cells, but then they're using the word cells again. I'm being totally pedantic, but that's just my thing. So they're saying human-induced pluripotent stem cells from patients with diabetes or other metabolic syndromes have been derived. And welcome back, Xeron. Thank you for returning. But this is an understandable oversight, even though this presumably got past peer review, or like multiple rounds, or multiple peer reviewers, and multiple rounds of people, editing it, and multiple iterations of the manuscript. Work in progress in our laboratory has demonstrated that this protocol can be applied to differentiate stem cell beta cells from diabetic human-induced pluripotent stem cell lines, which could be used for disease modeling and the study of beta cell function or susceptibility to stress or immune attack. Fair enough, you are correct. It's not. No, it's largely to, evaluate the science and the research and the quality of the research. But, ideally, they should be reading every single word in the submitted manuscript. But, I will fully admit, as someone who advocates for, significant reform of the peer review system, I know, and that's why, again, I guess as preview for a future stream, I will be covering this that was published recently, of like, hey, there's let's say more and more calls to replace our current publishing system, or as they say, starting off, replacing traditional journals with a more modern solution that is hopefully a significant improvement. That remains to be seen. Where was I? Yeah, work in progress in our laboratory has demonstrated that this protocol can be applied to differentiate stem cell beta cells from diabetic, human-induced pluripotent stem cells, which could be used for disease modeling and the study of beta cell function or susceptibility to stress or immune attack. Furthermore, technologies like TALEN and CRISPR, which I think virtually everyone knows about, let's say, the gene editing technology CRISPR, but there's this lesser known, although I think it's kind of like fallen off the map. I don't think it's, as far as I can tell, but I haven't looked closely. But I don't think there's a lot further development of TALENs. But this is another gene editing technology that let's say at once, it was a big deal, but it seems like it's been since overshadowed by CRISPR. And I think virtually everyone has heard of CRISPR. I think very few, outside of the the research biology world, have heard of TALENs. But yeah, they enable genome editing of these other stem cells or human-induced pluripotent stem cells. They're doing it again. Same exact mistake, some redundancy. To test variants identified by genome-wide association studies. Stem cell beta cell drug responses could be compared between genetically matched pairs of mutant and non-mutant beta cells. Identification and testing of novel biomarkers for beta cell function for pharmacogenetics is also enabled by the combination of these technologies. Thus, stem cell beta cells provide a new platform for in vitro drug discovery and characterization for human metabolism and diabetes. So, as they close out, the generation of stem cell beta cells provides a potentially useful step toward the generation of islets and pancreatic organs. Incorporating pancreatic niche cells, such as mesenchymal or endothelial cells, into cultures of stem cell-derived pancreatic cells may be beneficial. Other evidence suggests that the presence of alpha and gamma cells, that is the gamma, I want to make sure I have my Greek properly. Yes, that is gamma. Yeah, gamma cells may be important for precise tuning of normal beta cell function. Tissue engineering of a pancreatic organ will require incorporation of exocrine and ductal tissue, possibly in a specialized architecture. Much work remains to be done to achieve any of these therapeutic, disease modeling, drug discovery, or tissue engineering goals, but the generation of stem cell beta cells represents a step toward making a clinical impact with stem cell biology. That is a fantastic question. because that is one of the major hurdles, as highlighted here. But in the therapy, this therapy that was just recently FDA approved, that is like one of the challenges, where like some patients, they require immunosuppression, so that they don't destroy these foreign injected cells. But it's potentially possible, okay? Given the fact that they are starting with, human, or rather they're starting with, human-induced pluripotent stem cells, okay? As they say here, human pluripotent stem cells. It might, I'm not saying this is for certain, this is just like me thinking out loud, take what I say with a grain of salt. But there may be some potential that they can take cells from you, you the patient, and they can essentially engineer them, or get them to develop into beta cells. And that should presumably, let's say, allow it or open the door for your body to recognize it, as, your own cell, or pretty much to not recognize it as foreign. Yeah, a sort of like broad overview. Probably not like I take this is just like for the sake of example. Probably wouldn't be a skin cell, but like, yeah, pretty much in principle, that would be like the logic of like taking a cell, some sort of cell from your body, okay, that it's already recognized as part of you, and then getting it to develop into a beta cell, or maybe super ambitious into a fully formed new pancreas. Maybe that's like super ambitious. I'm not saying that will actually be the case, of like that will actually be achievable, let's say tomorrow, or even like 5 or 10 years from now. But like on paper, there is some feasibility, or possibility there. And I do know, okay, for other stem cell therapies, for other stem cell therapies, 1 second. One moment. So this is like, one of the primary differences, or like, stem cell therapy, broad category. And I'm definitely not an expert on stem cell therapies. But that's one of the major distinctions when it comes to stem cell therapy, is like, allogenic versus autologous. Where like allogenic, so allogenic, it's like stem cells from someone else other than the patient, okay? But there are autologous stem cell transplants, of like they use the own patient's cells to develop the therapy, which presumably should have much less chance, if not virtually no chance, of being rejected. But I haven't like looked into the research for every single application. But like, again, in principle, there is some feasibility or possibility there, where they might be able to take your own cells, which should have much less chance of being rejected, and get them to develop into a beta cell. Fully formed new pancreas would of course require a whole pancreatic transplant. let's- yeah, well, so that's where it might get so, let's say, science fiction that it might just be, okay, that they inject you with, let's say, a few cells, or it's like an injection, a much smaller injection, and then from there it develops into maybe either a fully formed pancreas, or maybe close to a fully formed pancreas, or like somewhere in between, because that's essentially what they do with this therapy, this approved therapy. So like they're not replacing a full pancreas. They're essentially just trying to replace a specific cell population, or pretty much they're trying to replace specifically insulin-producing cells. And there appears to be some effectiveness for some patients for some amount of time. And maybe, not saying this is the case, but maybe somewhere down the line, we can start from an injection of cells, and it might develop into maybe something close to resembling a pancreas, or like maybe the most ambitious, a fully formed pancreas. I mean, that's probably much less realistic, and probably a much larger timeline. But again, there might be some possibility there. When I became diabetic, my body was attacking its own beta cells. How would a stem cell turn beta cell from a... Right. So that's an excellent question. And that's why I believe that's one of the reasons why, for some patients, they require multiple infusions. And so it's a valid question because, like, at the end of the day, and actually, okay. So I'll use my own disease as an example. Because stem cell therapy is, let's say, multiple avenues of stem cell therapy is being used to treat, my own disease. And so this is autologous, okay? So they're trying to use the patient's own cells. Okay, so one of the, way back in the day, okay, and this has sort of been tried, of like, so I have, what's very often referred to as an autoimmune condition, even though technically speaking it's not, or like the most recent research points to the fact that it's not, but whatever. It's an inflammatory condition, and that my condition results in, the destruction of parts of my GI system. And so it was proposed at one time, where it's like, hey, why don't we just like replace parts of their gastrointestinal tract? Of like, hey, if the disease is destroying the small intestine, well, how about we just transplant a new intestine? But then, as you would probably correctly point out, and was, let's say, research or experiments have pointed out, well, just the same process happens over and over again. Like, well, yeah, if its origins are in the immune system, and it's just going to, attack the digestive system, well, then you're providing more food for it to destroy, or like more stuff for it to destroy. So it's a fair point that, like, in the case of type 1 diabetes, yes, they are just, you replace the beta cells, but the overall, like, the root cause is autoimmune, and your body's own immune system destroying those cells, well, then you might just be on the same trajectory. Like, well, yeah, you can replace the beta cells, but they're just going to be destroyed again. Which is a totally valid concern, and totally a valid question. So, one of the potential ways that I can, this is me thinking out loud, one of the potential ways that I can see sort of addressing that issue, because this strategy has been, is sort of like being trialed, is like, yes, as you correctly point out, so multiple avenues of restarting the clock. Either restarting the clock of like, hey, you get a fresh set of beta cells, like they will be destroyed, okay? But maybe it'll take like 2 years. Oh, and TransFemcell, thank you so much for the follow. Much appreciated. Happy to have you here. I think I've for sure seen you around the Twitch politics space. But yeah, thank you so much for passing by and the follow. Super appreciate it. So yeah, so there's multiple avenues of resetting the clock. It could be that you get a new population of beta cells, and it might just take years. I don't know how long, okay? Maybe, you know, I can see where you might just need an injection every 6 months. It's like, yeah, the beta cells will be destroyed, but hey, as long as, you know, it might just be like an annual injection, or like a couple times a year, or like maybe most ambition, like every like 3, 4, 5 years. Technically not a problem, or like much less of a problem, if, you know, you have a decent supply, or you can make these beta cells with, you know, they're accessible, and that's what that research that I just covered points to. Of like, they're saying like we might have an unlimited supply or close to an unlimited supply of beta cells, if in fact we can push this to, you know, fully function human beta cells that can be injected. And yeah, as you say, much better than 4 injections of insulin a day. But then like the most ambitious, okay, potentially, and this is the research that I have on screen, is like, well, it could be both an injection of beta cells, maybe combined. This would be considered very aggressive. But this would be sort of like resetting the immune system. So this is one of the strategies that has been tried with my own condition, Crohn's disease, where it's like, you know, it originates in the immune system, and we attempt to reset the immune system, where it's a very experimental and very potentially risky procedure, because through resetting the immune system, through stem cell autologous hematopoietic stem cell transplantation, through on the process of doing that, we pretty much wipe out the current immune system of the patient, and so they're much more susceptible to like infection, and they pretty much, you know, they have to be isolated, and like, you know, put on antibiotics and a whole bunch of things to make sure that they don't get an infection that will like kill them as we're resetting their immune system. Oh, and Dorcas Prime, welcome. Thank you so much for passing by, and good to see you. What would the pricing theoretically be compared to insulin? Because for people, it's 100%, and I can totally see this potential, like, not there yet. At least this particular fully in vitro is not there yet. But I would suspect, at least given what I know of, let's say, cell therapies, or like, you know, somewhat similar cell therapies, or just in the broader range of biologic therapies, probably pretty expensive. Probably on the order of thousands of dollars, if not tens of thousands of dollars. But at the same time, it's always like cost-benefit analysis of, like, you know, type 1 diabetic patients. They often, unfortunately, but understandably, require, you know, fairly intensive and regular medical intervention for a variety of reasons as a result of complications, of, you know, of challenges in managing their blood sugar. And so even though this would be potentially expensive, I don't know for sure, but I'm reasonably sure over the long term, if this results in better control of their blood sugar, almost certainly there will be, over the long term, much lower rates of long-term complications, and that they would probably require, over the long term, much less medical intervention. And so I think, yeah, the initial upfront cost would be expensive, and And I don't know for sure, but I would project that over the long term, it would be an overall cost-saving therapy. But yeah, so like going back, of like, I could potentially see, if in fact this type of therapy, which is like very risky, and very kind of like invasive and requires a lot of time, resources, and patient screening. And even then, there's no real guarantee. But I can sort of potentially see that you might be able to reset the immune system of a type 1 diabetic patient. And then if we actually have the ability to replace their beta cells, their pancreatic beta cells, well then it might be curative or really close to curative for a type 1 diabetic patient. But that's like really, you know, aggressive type of treatment. Because again, resetting a patient's immune system is a risky procedure. Like as of now, as of today, like, quite risky and like very invasive. And that's why like this therapy is typically only reserved for like the most severe cases of ones that have like failed virtually everything, and they're like usually like screened quite heavily that they can actually like survive the treatment or therapy. And even then, it's no guarantee, because we're sort of like finding out, at least when it comes to my condition, that even if when we try to reset the immune system, like a full reset, it seems to come back for some patients. Sometimes it comes back after a year. Sometimes they get like 5-10 years. But that's why they say among adult patients with refractory Crohn's disease not amenable to surgery, and like their quality of life is like, you know, really bad. And like, yeah, at 1 year is associated with significant toxicity. And so it's like, it you know, for some studies, this type of really aggressive invasive treatment has shown some promise. And in other studies, like this one, like at best, it's mixed. I'm also curious if this is something that might be covered under Quebec's healthcare. Well, so it's a valid question. As someone who's in Quebec and receives Quebec healthcare. Second. I would say the chances are quite high. Given the fact that, okay, like, so for my own therapy that I take, and I take an injection every month. Yeah, every month. It costs around $5,000, or like more, to be more specific, like $4,800 Canadian, and it is covered by the Quebec government, thankfully. And so, given the fact that I'm reasonably sure that, again, if this actually comes to market or results in an actual approved therapy, I'd be willing to bet that this would be cost-saving over time. It would reduce the rates of hospitalization, or I don't know for sure, but I'm reasonably sure it would reduce rates of hospitalization, it would reduce rates of long-term complications, and so it would be in Quebec's, you know, long-term interest to cover it. And like, I'm pretty confident that they would. Again, it would have to be rigorously tested, and some sort of analysis would have to be carried out. But I'm pretty confident that it would be approved. Wouldn't resetting the immune system essentially lead to either infections or more aggressive? So that is exactly correct. At least during the process of resetting the immune system. Where, as mentioned, for therapy like, for therapy like this, you know, the autologous hematopoietic stem cell transplantation, they have to wipe out the immune system. And then they, you know, transplant the new one, or like, yeah, they transplant sort of like whatever, the seed for the new one, and then the new one, you know, grows and develops. So like during that process, as they wipe out the original immune system, there's increased risk of infection. But like, yeah, once the new immune system takes hold, I don't know if I haven't looked at the data, but I don't think there's a greater risk of infection. It seems like you know, the immune system functions pretty well from what I can tell.
Welcome to Flavor Country.
And Kissing Gonzo, thank you so much for the follow. Much appreciated. Happy to have you here. Where it's like, yeah, it seems like it functions, assuming, okay, it's really tricky. But when it comes to like infections, it seems like it still, responds to infections decently well. The only potential issue is, or not only, but one of the potential issues but this is typically seen with a bone marrow transplant called graft versus host disease. Where it's like, I'm not sure how common or if this is even an issue with autologous stem cell transplantation, because the stem cells are coming from the patient, so they should be recognized, by the original person, because the cells are coming from the original person. As opposed to for like a bone marrow transplant, like there's a donor, and so pretty much like essentially the person, the patient, is kind of like getting, is basically getting a foreign immune system, and like whatever, if it's essentially a foreign immune system, like pretty much it's going to recognize the entire patient as like foreign because it didn't originate from them, and it results in situations, well, that's why it's called like graft versus host disease, of like you're getting someone else's immune system, it's just being transplanted into you, and now like pretty much all of you is seen as foreign because it's not your original immune system, literally pretty much someone else's. And a bunch of therapies have been developed or are being developed to treat this. But in a case like this, I don't think that's an issue. And it seems like, if you can actually get through all the hurdles and challenges associated with a therapy like this, which is quite invasive and quite aggressive, it seems like the immune system behaves quite well. I don't think like perfectly normal, but I don't think from the data that I've seen, does not appear to be a much greater risk of infection or whatnot. Yeah, so that's what I wanted to cover for diabetes. Obviously I went on a bunch of tangents and helped myself. But the other thing, the last thing that I want to cover, kind of like a brief story that I think is pretty cool, that I can relate, kind of like my own personal experience. So that is a fantastic question. That I don't think we have firm answers to. So, I think there's a lot of, as someone with kind of, let's say, either autoimmune or autoimmune adjacent condition, and as someone who is taking advanced immunology courses, and like reads the research on immunology, and I think has pretty good working knowledge of the immune system, but not perfect. I'm not an immunologist. But like, there's this kind of like concept of, let's say, immune training or like priming of like, what allows an immune system to develop, and like, what generates a strong immune system. There's some people think that, you need to be as you're growing up as a child, you need to be exposed to all these things in order to train your immune system to, to develop a robust immune system. And that, some people go, there's like some sort of camp out there that like will advocate of like, your child like eating dirt and like intentionally exposing themselves to stuff as like a way to train their immune system. I don't think the research bears that out, and I know, or rather, I'm quite confident that the recommendation that your child should, like, play in dirt and eat dirt, I think is explicitly unfounded. But all that to say, it's like, seems like your immune system needs some kind of training, but where that training is coming from, is it coming from, the bacteria that's in your body? Oh, Nabil, welcome. Thank you so much for passing by. And also shout out to Nabil, fellow streamer, that I believe they were doing a fundraiser just recently, that I hope went well. I have no doubt that went well. And thank you so much for passing by. But everyone should be giving Nabil a follow. They do great content. Absolutely. All hail maple syrup, especially as a Quebec resident. Like, that's one of our primary exports. I think it's pretty much just like poutine and maple syrup. Oh, that's fantastic. I'm super happy to hear that. That's fantastic. And I'm glad to hear that it went well. And like, I'm hoping one day I can get to the point of doing my own fundraiser for causes like that I care about. But yeah, where was I? Oh yeah, so like, yeah, immune system training or priming. Of like, yeah, it does seem like some sort of training is required. But I think there's still a lot of open questions of like, how much training, where is it coming from? Is it really through exposure of like, infections or childhood infections, or like, just like in an unsanitary environment? That doesn't seem to be the case. Or at least I don't think the research so far supports that hypothesis. But then like, okay, what about like, your microbiome, or like, parasites that maybe at one point we were exposed to, but due to the fact that, at least in developed countries, we have, much lower rates, or like parasitic infection is pretty uncommon, if not rare. And so it's like, well, maybe some of that immune system training is being lost, since, maybe we're too clean. And there have been some potential therapies that have been explored to actually address just that. Again, I'm going to come back to my own condition as an example of like one of the treatment avenues that have been explored is like actual intentional parasitic infection as a treatment for my own condition, using specifically this is old, outdated. It's essentially been abandoned. Yeah, this goes back to 2005. But basically, the overall thought process was, this is like going by like the hygiene hypothesis of like, hey, given the fact that, my own disease appears to be way more common in developed countries, particularly in Canada, we might be too clean, and that maybe eliminating all of the parasitic infections that we were exposed to, not a good thing, and results in conditions like mine or other autoimmune conditions. So like way back in the day, even earlier than 2005, it was like thought that, well, what if we intentionally infect patients with parasites, and that maybe that will have some sort of immune modulating effect. Well, spoiler alert, this has essentially been abandoned, and like it never seemed all that promising. There was like pretty much the research was looked promising at one point, but then upon further research, it was like, oh, there's like nothing really happening here. Oh, well, yeah, now I'm actually glad to hear that. I think we should start poutining just about everything. I think pretty much just about every dish is improved by poutine. And that's one of the things that I love about Quebec, is pretty much all of the different varieties of poutine that I have access to. That is like, yeah, super great. And even though I have a lot of criticisms about Quebec and how whatever Quebec is run and its politics, I appreciate and I'm grateful for, let's say, it's all of its varieties put in. That there's pretty much new ones coming out, and I have not heard of the butter chicken one, but it sounds amazing. And yeah, so that is my understanding as well, where pretty much this type of, let's say, therapy, at least when it comes to trying to modulate the immune system, has been abandoned. Probiotics, yes. I think that's pretty much an active area of research that I think we're still trying to, like, tease out. As it's like, again, this is all, like, emerging research. The concept of the microbiome and probiotics is, like, pretty new for, like, only in the past, like, 10, 15, 20 years. And I think a lot of the research, or, you know, a lot of the claims about probiotics, or at least when it comes to, like, what's marketed in a, like, in a supplement store, I think a lot of those claims have been overhyped. But there are some actual therapeutic probiotics that have been developed, or let's say pharmaceutical probiotics that have been developed. But there's still a lot of unanswered questions. And same thing with fecal transplants, where, again, over and over, I come back to my own condition of, like, fecal transplants. They appear to be effective for ulcerative colitis, and they pretty much don't seem to be effective at all for specifically Crohn's disease. Whoopsies. Where at least a lot of, quite a number of trials have been attempted to see if fecal transplantation is effective for Crohn's disease. So far, of the ones that I've seen, or pretty much all of the ones that I have seen published, you pretty much don't get any real positive results. Seems to be effective, or there's some effectiveness for ulcerative colitis, but when it comes to Crohn's disease, there's pretty much, so far, it has fallen flat on its face. So far. Still being tried, and I know there's still trials being conducted to see if we can get it up to actual therapeutic benefit. Because, you know, we're, there's still a lot of unanswered questions in terms of, like, well, okay, you know, who do we get our fecal transplantation from? You know, how much do we give? How frequently do we give it? Etc., etc. Where it's like, yeah, since we pretty much, I would say there's way more unknowns about the microbiome, especially when it comes to, in the case of, you know, Crohn's disease patients or ulcerative colitis patients. It's like, yeah, so many unanswered questions. But, like, yeah, I think you are correct, and that's my understanding as well of, like, yeah, parasites in general, or, like, you know, intentional infection with parasites has been abandoned, and so far, like, yeah, most of the avenues of research are, like, exploring probiotics and fecal transplants. And I think as of, like, today, at least in North America, like, fecal transplantation is only approved for the treatment of Clostridium difficile, or, like, the bacterial infection, C. difficile, which it seems to be actually really quite effective there. And I think that's pretty much, if I remember correctly, that's the only thing that it's been approved of so far, since a lot of the research is mixed in other applications. But yeah, all that to say, where it's like, yeah, some sort of training of our immune system seems to be required, but I don't think we quite know, like, how much training, like, and it probably doesn't need to be super invasive training of, like, you know, intentionally infecting someone, like, with a parasite, or, like, you know, having your child, like, eat dirt or something. Yeah, I think that's probably, like, the most realistic and probably like the most research backed. Of like, hey, you probably don't need to like intentionally seek out infections. You might just need, you know, a boost to your microbiome. Even though a lot can go wrong with our bodies, obviously, that, you know, I'm highlighting now, I think overall our bodies are pretty resilient, generally speaking. And that, you know, I think it's pretty safe to say that, you know, intentionally seeking out infection almost certainly will never pan out as like a good strategy. I think I'm gonna save this for my next stream, as I think everything that I wanted to cover as a main topic has been covered, and I think this will take a little bit longer to cover, even though this is like a brief story. But I have other research that I want to bring in that I think makes this cooler. So yeah, I'm gonna end the stream here for today. I want to thank everyone for tuning in. Let's see if I can find someone to raid. And so I should be back on Wednesday. More, I'm gonna take a day off. But like, yeah, given the fact that my sleep is holding up, and I'm pretty confident that it will, hopefully forever, for at least the next little while, I will be back. I have every intention of being back on Wednesday. And I'll save this, and I have a whole bunch of other content that I want to cover on Wednesday. But I'm probably gonna start off with this. And research related to this. Like, you know, there's other published research, pretty much right up my wheelhouse, showing that, hey, maybe you can get a lot of information from a drop of blood. Maybe not, you know, Theranos, Elizabeth Holmes types of information, but some information that is actually legitimately research backed. Oh, and Jack, thank you so much for passing by. Super appreciated. And I'm sure I'll see you around the Twitch space. Definitely on a panel at some point in the near future. Let's see if there's someone to raid into. Wick is streaming. Gonna send you over to them. Rather him. Speaking of panels. Set that up. But yeah, thank you everyone for passing by. Dorkamus Prime, super appreciated. Jack, super appreciated. Nabil, and also shout out to you. Shout out. Lots of shout outs. Shout out to Johnny Scarlet for the raid, super appreciated. Johnny Scarlet with 2 T's or 1? It is with 2 T's. Follow both of them. Super appreciated for the raid, and thank you for the follows, Admiral Love. Thank you, Transfemcell, for the follow. Kissing Gonzo. And anybody else that I forgot, I apologize. Thank you, Zeron, for passing by. And yeah, anybody else that I forgot. Oh, and Tetsu Paul. A whole bunch of people to thank. But yeah, I'll be back on Wednesday. It'll probably be a little bit earlier. Or rather, a little bit later. By around 6, 7 p.m. Eastern Time. And also, yeah, shout out to you, Tetsu Paul. Another great Canadian. And Poutine lover, most importantly. And fitness streamer. Definitely follow them as well. So Wick is in the middle of doing a panel. This is not science and tech. This is not fitness. This is basically, this is debate bro politics that I occasionally dabble in. But yeah, until next time, take care, be well, and be kind. And if you're not following Wick, you should be following them, even though I don't agree with all of their positions. They're kind enough to have me on their panels. They're doing now. And if it's up your alley, give them a follow. But yeah, I'll be back on Wednesday. And until then, like I said, take care, be well, be kind.
That with respect to, I mean, we've talked already a little bit about how